everyone, and welcome to our webinar. My name is MJ Kim, and I'm with Mission Bio, and I'm really thrilled today to be your host today. So before we get started, I just have a couple of housekeeping items that I'd like to go over. This session is being recorded, and the link to the recording will be sent to you afterwards. And if you have any questions during the presentation, please type them into the Q&A box anytime during the webinar, and we'll have time for a Q&A session at the end. Today, we have a very special presentation on single-cell multi-omic characterization of multiple myeloma. And we're excited to have our very own Chief Technology Officer, Adam Shamby, as our speaker. Without further ado, I'd like to pass the floor to Adam.
Hi, everyone. I'm excited to talk to you today about our single-cell platform and how it can analyze multiple myeloma. Just got back from EHA last week, and it was, got a good practice run talking to everyone about this. See some of you on the list, and yeah, excited to go over what we can do with our platform on multiple myeloma. And there are two angles that you can look at when analyzing multiple myeloma. And represented in the title of this presentation. One is sort of understanding disease progression, and then also understanding getting some sort of therapeutic guidance for multiple myeloma from our platform. I should emphasize that our platform is research use only, but I think there's a lot of potential here. So the webinar is going to be divided into three parts. We'll have an intro into multiple myeloma, and specifically how, as a disease, it really needs to have clonal analysis to understand, to diagnose, and to treat. Then we'll talk about the Mission Bio platform, its DNA and protein analysis, and how it can meet the needs of multiple myeloma. And then as a third section, I'll present data, fairly new data, from a multiple myeloma study, specifically looking at paired pre-diagnosis time points, MGUS and smoldering multiple myeloma, and diagnostic time points, and how we were able to assign a diverse set of molecular attributes to subclones within that sample. And also understand, as I said at the top, maybe why the disease progressed, and also how therapy might be guided. So multiple myeloma, as I'm sure many of you are aware, is a clonal expansion of plasma cells, and it's a tricky disease because it often relapses. Usually, there's the first round of treatment seems to go pretty well, and there's remission. But often relapse occurs shortly after, and you require many rounds of therapy. And this failure is likely due to the clonal heterogeneity of the disease. There are lots of different clones with different variants associated with them. And when you treat some of them, but not all of them. At the same time, there might be variants associated with resistance to treatment, and that also would cause treatment failure. As I said in the intro, in this talk, we'll be talking about two sets of time points, the earlier MGUS/smoldering time points, and also the full multiple myeloma diagnostic time points. And so here is just a sort of depiction of what we'll be talking about. The two earlier time points with no symptoms and fewer signatures, and then the diagnostic time point multiple myeloma, which has more symptoms. And we'll be comparing these two at a molecular level. So multiple myeloma is, again, very tricky because it has many different drivers and indicators. On the left, you see all of the point mutations and that can be associated with, and driving genes that can be associated with multiple myeloma. And then on the right, you can see all of the structural variants. This is specifically looking at copy number that can be associated with more advanced disease. You can see there are many copy gain genes associated with, or chromosomes associated with copy gain. This is often referred to as hyperdiploidy. The odd chromosomes have gain. You can see that there are these associated with loss, and then various focal copy numbers. There are many, driving molecular signatures of multiple myeloma, and then there are also many treatments for multiple myeloma. And you can see here are some of the markers and various therapies from CAR-T, bispecific, monoclonal antibodies, ADCs, and inhibitors that can help treat the disease. You can see on the right here some of the drugs associated with this. So there are a lot of therapies, but of course these therapies can also be stopped by various mutations that then alter the therapeutic targets so that they don't work as well. So these are all things we want to understand. All right, so I've talked about some of the signatures of multiple myeloma, the molecular signatures. Let's talk about why there's often failure of treatment or relapse. This is a nice plot that represents bulk sequencing from a particular multiple myeloma patient, and how through several rounds of treatment, different clones were brought to the fore because of resistance. So at the diagnostic time point, you can see the various clones represented by these geometric shapes associated with this clone. And then after treatment, another clone became dominant. Here, clone 2, this is the first relapse. And then a different treatment was performed. Then this clone 1.2, which is actually more similar to 1.1, then came up. And then lastly, clone 3 came up. So it's sort of a game of whack-a-mole where you treat one clone and then another one pops up. What you'd really like to have a sense of is what are the clones at the start of treatment, so that you have a sense of which ones are likely to succeed. That's usually clear from bulk analysis, but then also have a sense of what's going to come next. And are there treatments that can span multiple clones? So just to reiterate that, the diverse clonal architecture makes for challenging therapy selection because you need to be able to treat many different clones, which have potentially mutually exclusive therapeutic targets. And really what you need is you really want to understand clonal heterogeneity. You want to understand it across a lot of molecular signatures to both understand the disease and then also design treatment. So what I'm going to do now is I'm going to walk through a more complete list of potential molecular signatures that we might want to look at, so that we can maybe better track disease and understand it. So here's a wish list of molecular signatures one might want to look at for multiple myeloma. And then I will give you some examples of each of them and say how they potentially are prognostic markers or maybe therapeutic markers. So first is clonal architecture. This is sort of the most important. You really want to understand what are the clones there in the patient, and where, and how are they spread? Is it a single linear evolution? Is it a branching evolution? And what are in each of those clones? You want to know clonotype, the BCR BDJ signature to understand clonality, and that often is one of the earlier mutations, and so that sort of helps you distinguish healthy sample, wild type sample from the mutants. Structural variants are very important. Multiple myeloma is a disease of genomic instability. And so understanding structural variants both helps you identify clones, even if they're not driving mutations, but also there are a lot of prognostic and therapeutic targets associated with structural variants and copy number. You will understand point mutations. Point mutations are really important drivers of multiple myeloma, and also therapeutic targets. Zygosity is very important because a homozygous mutant is often more deleterious than a heterozygous, but maybe it's also easier to treat because it's more responsive to inhibitors that target it. You want to understand immunophenotype and therapeutic markers to both, because this gives you prognostic sense of the disease, and then also gives you potential therapeutic targets. On the previous slide, I showed a lot of ADCs and CAR Ts and antibodies that targeted specific markers. And then lastly, you really want to understand in clonal dynamics, how is the disease changing and evolving? So what I'm going to show you now is that's sort of the wish list. Now I'm going to talk about some prognostic markers associated with each of these. Sorry that I'm running through a laundry list, but the punchline will be is that we would like to be able to analyze these all together, all in the same clones, because that will help you understand how one might want to treat it. So from clonal architecture, a prognostic indicator, one of poor prognosis, is if you have a very branched clonal architecture. If it's very linear, that's maybe better. If it's very branched, then there are many different competing clones, any one of which can become the next relapsed clone. In terms of clonotype, if you are in remission, a prognostic marker might be an MRD positive clonotype, a rare clonotype that was detected at diagnosis that you're now seeing reappear. For structural variants, there's loss of 17p, which is associated with TP53, loss of 1p, gain of 1q, and so on, hyperdiploidy, translocations, those are all prognostic. For point mutations, you have TP53, RAS, MYC, DIS3, and many others. For zygosity, as I said, you really want to know, is it homozygous? For immunophenotype, if it's CD56 or 138 or 200, those are all indicative of poor prognosis. And obviously, clonal expansion gives you poor prognosis. So this is a big list, but what is important to understand is not just are these present in the patient, but in a clone, which clones have which of these? Because they could be mutually exclusive in different clones, or they could appear in the same clones. So these are prognostic markers. And then similarly, there are therapeutic markers, which aren't too different from the prognostic markers. But from clonal architecture, it's therapeutically guiding to know that your target that you might have gleaned from flow or from bulk NGS is present or is not present in all of the clones. If there exist clones in which it's not present, that clone might be the one that causes relapse after treatment. And then we can go down the list. For clonotype, if you have a refractory clonotype, that probably means your therapy is not working. Structural variants, if you have biallelic deletion of BCMA, that might mean that your BCMA therapy might fail. For point mutations, there are a couple therapeutic targets: BRAF, V600E. As I said earlier, a BCMA resistance mutation again will probably indicate that your BCMA therapy might not work. Zygosity, a homozygous mutation actually might mean that it responds better to therapy targeting that pathway because of the reliance of that particular cell on this mutation to propagate. If you have heterogeneity of BCMA or GPRC5D or CD38, these are all therapeutic targets. That might mean that your therapy might not work as well if your assumption is that all of the mutants express these targets. And then lastly, for clonal dynamics, a therapeutic indicator of this is if you have, resistance. You see clones that aren't responding. Many clones might respond, but some might not be, and that would be an indicator that your therapy is struggling. So I've just, thank you for listening to this long list. There are a lot of things you want to understand in multiple myeloma, a lot of action that you can take from them. How are they measured currently? So here's that same list, and now I'm just going to list some of the technologies that can look at them. One of the most versatile is probably NGS. The problem with NGS is it can measure a lot of these things, but clonal architecture and clonal dynamics, you're mostly inferring from bulk. And then clonotype, structural variants, point mutations, zygosity, you can measure it, but it's not really single cell. So you can't tell which clones, how are these populations grouped together? Are some of these features mutually exclusive from each other, or are they co-occurring? Karyotyping in fish is another important way to analyze multiple myeloma samples. It's single cell for sure, but it's sort of limited in what it can look at: structural variance and zygosity. Very important for diagnosing samples, but sort of limited in what it can look at. Flow cytometry is similarly very powerful, but again, it's limited to protein, whereas karyotype and FISH are more to the structural copy number side. And lastly, you have single cell expression, which is more of a research tool, but you can, again, infer clonal architecture, infer clonal dynamics. You can look at clonotype and phenotype, but there's no real platform that can look at all of these together. And so, it's really important to understand, because what if you have clones in your multiple myeloma that are mutually exclusive, that have features that are mutually exclusive from other clones? So now I'm going to talk about the Mission Bio platform very briefly and sort of show you how we can come in. So I'm going to take those same attributes here. I'm going to group them together a little bit. These are the same ones. And now I'm going to show you what the output of the Mission Bio platform looks like. So I've got this rather busy table here in the center. And you can see that there are many rows, and each row corresponds to a subset of these attributes on the left. You got clonal architecture. You can see that the clonal branching. You can see VDJ here. You can see the structural variants, some of our favorite ones: hyperdiploid, deletion of 1p, deletion of 17p. You've got arm level deletions and gains. You've got counts. You got point mutations. You have phenotypes. CD19 and 45 are more typical of healthy cells. Then you have CD56 and 138 and BCMA and FcRL5. These are all prognostic and therapeutic targets. And then you have, for each row here, you have the population size at different time points. For each column, you have these clones. By bulk, this is all blended together. But now we can resolve these subclones, these different genotypes and phenotypes within the patient. And you can see these are very different. Clone C2 here is hyperdiploid, has a lot of arm gains. You can see that those sort of track together, and has deletion of 6q. You scan down, and it seems to be negative for BCMA, but positive for FcRL5. You can see from the MGUS at diagnostic time point that the sample seems to be, the clone seems to be shrinking. Clone 6, if you scan down, it has gain of 1q. It has a homozygous NRAS mutation Q61H. That's what the red box means. It means homozygous. And that seems to be the one that's growing. It seems to have higher BCMA expression than the wild type, but maybe FcRL5 expression on par with the wild type. So already we're sort of getting a sense of the complexity of this sample by looking at all of these attributes. So that's sort of a primer to multiple myeloma. I'm now going to breeze through some of the single-cell DNA analysis, our single-cell DNA analysis platform, sort of how it works, and then we'll get on to the multiple myeloma study. So Mission Bio, how are we able to do this? Why do we do this? First, let me say that single-cell DNA is valuable. I think I've already been sort of emphasizing that, but it's really important to understand, are mutations occurring in the same cells, or are they occurring in different cells? Single-cell DNA is really hard to analyze in a scalable way because DNA is wrapped up in chromatin. Chromatin, you can fit a meter of DNA in a cell, but it also makes it hard to analyze in a sensitive way in a scalable small reaction. So RNA is relatively easy to access from cells in small volumes, often used to analyze single cells, but chromatin is very hard. That said, Mission Bio has some technology that has enabled us to first digest the chromatin in a very small volume and free the DNA, and then inactivate the digestive agent enzymes so that you can then perform single-cell, single-molecule sensitive PCR. And that enables you to detect zygosity, which requires the sensitivity of a single molecule, 10,000 cells at a time and thousands of targets. Here are just some of the papers. We have over 100 publications associated with our platform. These are all oncology based. And then here's how the chemistry works. So you start with a cell with got DNA and protein in it. You apply an antibody tag that tags the protein with an oligo. You then put the cell in a droplet on the bottom left here. You put the protease in. The protease, as I said, or as I referenced earlier, digests the chromatin. So now you have just DNA and your antibody tag. You then load it into a droplet with a barcoded bead. That barcoded bead labels every single molecule associated with that cell, but you can then sequence downstream. And so now you can, from our platform, identify by single cells which genomic DNA and which antibody, and therefore which antigens were associated with the cell. We have, you can think of us as a sample prep solution for NGS, but we enable everything around it to get you to your solution. We help you design targets. We have reagents and a cartridge that processes your sample. We have an instrument that runs the cartridge. And then we have analyses that help you interpret what comes out of the sequencer. For multiple myeloma, here are the targets, the DNA targets associated with our offering. You can see there are 34 genes with hotspots. We have 13 genes with various resistance mutations. We look at various focal copy numbers. Those are 10 genes. And then we have our VDJ clonotyping panel to help clonotype the cells. We also have a whole genome CNV panel with resolution that's 5 to 15 megabases. You can see here are the targets across the genome to help you get a good sense of arm level copy number. So I've listed a lot of attributes. One attribute that we are still working on, but I want to be upfront about, is that we cannot cover translocations. The 414 translocation and 1416 translocations are often very interesting for multiple myeloma. We can't cover those yet, but those are usually earlier mutations that can, or variants that can be associated with clonotype or other such mutations. So I did want to be upfront about that. And then lastly, we've got our antibody cocktail to help phenotype and identify therapeutic targets of multiple myeloma. Here's our 19 antibody list. And I've just chosen one, BCMA. That's, a lot of therapies developed around that. And here's I just wanted to show a ridge plot showing how we can distinguish these 4 cell lines that were pooled together in one run, and how the BCMA positive cell line really stands out. All right, so that was a quick primer on multiple myeloma and also on the Mission Bio platform. And now I wanted to go through our multiple myeloma study, looking at the paired time points of MGUS smoldering to multiple myeloma diagnostic time points, and what you can learn from it. The study had 20 pairs of samples. They were CD138 enriched, and they were cryopreserved. Our platform works well with samples being multiplexed together because we can identify germline mutations. You can take up to 3 samples and put them together in one run, and then they can be deconvolved using their germline variants. So in these runs, each time point was multiplexed in groups of 3 with others. There's minimal crosstalk, and on average, we had 3,500 cells per time point. So on average, about more than 10,000 cells per run. And then they were evaluated with the panels that I showed on the previous slides. So jumping into it, we have this sample, which I referenced in an earlier slide. What I've got here is the clonal tree of this sample. You've got the earlier time point. In this case, it's MGUS. And then we have the later time point, which is 13 months later when the patient was diagnosed. And what you see is there's this branch structure. You start off with wild type cells with the diverse VDJ signature. The founding clone becomes clonal for VDJ, has loss of 13 and loss of X. Loss of 13 is prognostic. You can see that then branches into this very large clone, which is hyperdiploid, has copy gain and a lot of chromosomes. And that's competing with this sort of long vine here in this tree, where you have a sequential loss of several arms. At clone 4, we see that we have the acquisition of an NRAS mutation, Q61H. Clone 5, due to copy loss of 1p, that NRAS mutation becomes homozygous. Because you basically lose the wild type copy. And then clone 6 gains a 1q, which is another prognostic indicator. You can see from time point 1 to time point 2, clone 2 shrinks dramatically. I would say it's probably not shrinking, it's just being outcompeted by clone 6, which just has a tough combination of attributes. It's got this homozygous NRAS mutation, it's got loss of 1p, it's got gain of 1q. That makes this very fit. Here's that table that I showed earlier. But now you can pair it to the clonal tree. You can see clone 2 with this hyperdiploid. That's this here. You can see this big red dot indicating the hyperdiploid, the number of arm level gains. And you can see that it starts off large and then becomes small. That's the red dot. The red dot means that it's shrinking, or at least no longer being the dominant population. And then clone 6, which you see here, is the one that has the deletion of 1p and the gain of 1q. You can see how the NRAS mutation went from heterozygous to homozygous as 1p was deleted. And then scanning down, you see that it is growing. What I think is important to note are, as I referenced earlier, is that the prognostic protein markers, you see that clone 2, which is the earlier clone, it seems to be low for BCMA. So, if you were targeting clone 6, which is the dominant one diagnosis for BCMA, you might expect clone 2 to be the source of relapse, as this arm of the clonal tree recedes due to BCMA treatment. However, clone 2 is also highly expressing for FCLR5, which gives you another potential target. And so this is just reiterating that. Now let me read it out aloud. Clone 6 with a homozygous NRAS mutation and loss of 1p and gain of 1q outcompetes the formerly dominant hyperdiploid clone C2 from MGUS. The C6 clone should be more responsive to BCMA therapy, while C2 might be more targetable by CD200 or OX2 and FCRL5. So now let's move forward to another sample. And this sample is interesting because there are 2 child clones, one of which has an NRAS mutation and one of which has a KRAS mutation. So by our assay, the only difference between these 2 clones are those mutations. So it's very interesting. You can now pit these 2 clones head to head and see what happens. You see here that the NRAS mutation is the one that expands. It more than doubles in size, and that the KRAS mutation is halved. Coming over here, you can see that clone 3 and 4, which are those ones we just talked about. The only difference with them is the NRAS and KRAS mutation. And you can see that the KRAS mutation is the one shrinking and that the NRAS mutation is the one growing. Looking at this, though, we look at BCMA, we see that everything is positive for BCMA, as far as we can tell, which means that this is, even though there's this clonal diversity and that there's differential populations, BCMA is likely going to be a good target for you and for this patient. And then lastly, I have a third sample here. This one doesn't change much from time point 1 to time point 2. It was 9 months apart. The other time point was smoldering. But what's interesting is that some of these clones, if you scan over here, some of the are positive for BCMA, and these are likely going to be responsive, but that some of these other clones, all of which are not really changing over these 9 months, you might expect if you treat it with BCMA that the clones over here would shrink, and this clone might be left to proliferate. All right, so I just showed you 3 samples. We can put all of this together in a huge table, which is a little daunting when you first look at it, but what's helpful is to scan the bottom, and you can see these red dots and these green dots. And what you're looking for are commonalities between the green dots and commonalities amongst the red dots. And that helps you understand, these are pre-treatment samples, but you can imagine with relapse time points with some sort of therapy, this would help you understand maybe why a treatment across a cohort is working or is not working. Just in this cohort, you can see that NRAS Q61 is always tied to expanding clones. It always has green dots. The KRAS mutations also usually expand, except for that one case that I just showed you where it was competing with NRAS, and in that case, it lost out. And then I haven't shown it here, but in general, the more structural variants you have, the more likely a clone is to expand. So this is very exciting. There's a ton of information here. It really, maybe helps you understand why certain clones and certain diseases are progressing, and in ways that you wouldn't be able to observe with traditional technologies. I wanted to have one last sample to show you, which is not actually from the multiple myeloma indication. It's actually from AML MRD, but I think it showcases the sensitivity of our platform. So if you look at AML MRD, you have very, very rare clones that can be very important, especially at the remission time points if you're looking to see if things come back. Here I'm showing you the clonal architecture of a diagnostic AML sample, as well as the remission time point. And this remission time point is MRD negative by flow. We see this clone here, this FLT3 IDT, which is ITD, which is a treatable target. There's there are FLT3 inhibitors. We detect it at 0.3%. We enriched the sample for CD34-117 by duplex sequencing. This is detected at 0.025%. So with our protocol, we are able to detect cells that are present in 1 in 4,000. And we can characterize it, and look for therapeutic targets within those clones. So that roughly sums up our multiple myeloma study. I just want to leave you with some conclusions. Again, this is a research use only, but hypothetically, in the future, trying to think of what are some actions one could take from the data supplied by our assay. First, you might want to supplement a particular therapeutic target. If our assay says that there's a clone that either has biallelic loss of that therapeutic target, say you have biallelic loss of BCMA in some clones, and you're treating with BCMA CAR T or BCMA ADC, that should give you pause. If there are clones that are low expression of the target, those might be the ones that relapse. And then also, if there are resistant mutations in a particular target, for a particular target in a particular clone, those might be the cells that don't respond to treatment. Alternatively, if you have a mutation, you might want to target new pathways, such as a BRAF, if that clone is a homozygous mutant, because that clone is going to be both more pathogenic and also maybe more responsive to therapy. And you also want to prioritize these pathways if they are mutually exclusive from clones that have the primary therapy. So if you have cells that are pathogenic because of certain mutations, but don't express your therapeutic target, you might want to add an additional treatment to target that particular pathway. And then more broadly, if just in terms of risk stratification, If the clonal architecture is very branched, that probably makes this patient higher risk, just because there are so many, there are more potential candidate clones for relapse. And then also if there's really complex genomic instability, that sort of suggests that there's more opportunities for deleterious variants. And then just to summarize, so that's actionability of our platform, potential actionability. And then also there's just in general for our assay, I just wanted to leave you with some big takeaways. I've hopefully been able to convince you of the value of clonal architecture, both to predict disease progression, understanding not that you have to come away from this knowing that there's not one clone, it's not a homogeneous sample. There are many, and to understand how the disease progresses, you need to understand those clones. And then also there's the potential to guide therapy, because amongst those disparate clones, there will be different levels of therapeutic target, and maybe alternate pathogenic variants that will cause those clones to expand. We've also shown you some features or molecular attributes that only we can detect. They're very hard to detect by other platforms. We can detect low frequency copy number events, even below 1%. We can detect low frequency homozygous mutations. Those are both prognostically and therapeutically important, down to 0.1%. With our clonal platform, we can look for the co-occurrence or the mutual exclusion of various molecular attributes within those clones. We can tie those to prognostic and therapeutic markers, such as BCMA, and then we can clonotype these, which is valuable for both understanding the expansion disease, but also tying to other assays that are other frontline assays, such as clonoSEQ. All right, so I've talked a lot. I hope you enjoyed it. Thank you very much.